Hello, I'm Dr. Diveshri Gangaraju, heading the Bioprocess Research Division at IOM Bioworks Private Limited, Bangalore. In today's video lecture, we would be discussing about different characterization methods using which you can characterize the probiotic microorganisms and how these probiotic microorganisms are classified. Characterization methods are the set of procedures which helps us to evaluate the survivability of a given probiotic strain under the various conditions of gastrointestinal tract. Previously, we have discussed the different aspects of gastrointestinal tract. So we all know it starts with the oral cavity, which comprises of alpha amylase, which might have a profound effect on the viability of an ingested probiotic strain. And then we have a stomach compartment wherein the pH of the stomach compartment varies from 1.5 to 3. You also have pepsin and trypsin as the digestive enzymes, which might have an effect on the viability of the ingested probiotic strain. Once the probiotic microorganisms passes stomach, it reaches the small intestine. Again, the pH of the small intestine varies between 6.5 to 7.2. You have bile, which is a yellowish green fluid, which aids in the absorption of the fat molecule, digestion and absorption of the fat molecule. Even this might hinder the viability of the probiotic microorganisms. And once it passes the small intestine, it reaches the large intestine, which is reported to be a site of the probiotic microorganisms. So as these ingested microorganisms pass the different aspects of gastrointestinal tract, so all of these compartments will hinder the viability of a probiotic microorganisms. Therefore, it is very much adequate and necessary to determine how the individual compartment will affect the viability of a probiotic microorganisms. Let us discuss each one of these in detail as we passes the slide. Let us now discuss the stepwise characterization procedure in order to evaluate the viability and as well as the functional robustness of the selected probiotic strain under simulated gastrointestinal conditions. So this procedure, the stepwise procedure for characterizing the probiotic strain has been detailed in one of my earlier published articles in the journal Bioprocess and Biosystems Engineering in the year 2015. So as we all know, in the GIT, gastrointestinal tract, the mouth or the oral cavity represents the first compartment of the human GIT, and this plays a critical role not only in the mechanical and the enzymatic breakdown of the ingested food, but will also play a major role in shaping the initial exposure environment for the orally delivered probiotics. And in the saliva, the salivary alpha amylase is a key enzyme that catalyzes the hydrolysis of dietary carbohydrates. Importantly, the alpha amylase activity affects the viability and physiological state of probiotics during their transit through oral cavity, thereby influencing their ability to colonize or exert the functional effects downstream in the gut. To incorporate this dimension into your in vitro model, what you have to do is you need to expose your probiotic microorganism to the simulated salivary fluid containing physiologically relevant concentrations of alpha amylase. As you can see, in our case, we have used about 100 units of salivary alpha amylase prepared in zero 0.1 molar sodium acetate buffer of a pH 5.6, and the exposure of the probiotic microorganisms to the salivary alpha amylase solution was performed for 2 minutes, and then you evaluate what is the survivability post-treatment. As you can see, please refer to the blue bar graphs. The initial cell concentration is found to be above 12 log CFU per ml. After treatment with the salivary alpha amylase, the concentration is reported to be around log 10 CFU per ml. This in turn tells that the salivary alpha amylase will have a profound effect on the viability of the ingested probiotic microorganisms, and it is very much important to determine how this salivary alpha amylase will bring down the viability of the probiotic microorganism. Next, we evaluated the effect of acid and bile tolerance on the selected probiotic strain, as acid and bile tolerance are the critical determinants of their survivability as well as the functional deliverability in the human GIT. Following the exposure to salivary alpha amylase condition, the probiotic suspensions were subjected to a sequential incubation in the medium. The medium which we are using is known as MRS medium, as we have discussed in the previous lecture, would be adjusted to a low pH value varying from 2 to 1.5 to 3. As you can see, in this case, we have varied at 3 pH values, pH 1.5, pH 2 and pH 3, in order to mimic the gastric environment, and subsequently in the media supplemented with ox-gal in order to simulate bile exposure in the small intestine. The ability of a probiotic strain to maintain viability as well as metabolic activity under these stress conditions, both acid tolerance and the bile tolerance, were assessed via colony forming units and viability staining assays. These steps are very much essential to characterize the capability of a strain to survive the harsh transit through the stomach and the small intestine compartment, and thereby it reaches colon in sufficient numbers to exert their health promoting effects. Now the figure towards the left shows the effect of acid tolerance on a particular probiotic strain, wherein the pH varied at 3 levels, 1.5, 2 and 3, and you see how there is a variation in the survivability of a selected probiotic strain. At 0th hour, being exposed to a pH 1.5 or pH 2 and 3, the viability of this strain was reported to be around or between 9 to 10 log CFU per ml. But as the time progresses, with the pH value, there is a severe decline in the viability of the probiotic strain, and this proves that the pH plays a very important role, and this affects the viability of an ingested probiotic strain. The graph towards the right shows the tolerance of the bile towards the viability of the strain. And again, as you increase the bile salt concentration, bile salt, the type of bile salt which is used or which we generally use in the procedure is known as ox gall, which is a dried bile extract which comprises of cholesterol, phospholipids, bile salts, and even bilirubin, which is a byproduct of red blood cells, which might have an effect on the viability of a probiotic strain. And these bile solution, which is yellowish to green in color, basically produced in the liver and gets stored in the gallbladder, and when we ingest fat containing food, it gets released from the gallbladder into a small intestine, and upon its release, and if you have a probiotic microorganisms being ingested and if it is in the small intestine, the bile might have an effect on viability of a probiotic strain. And the bile concentration in the small intestine might vary between 1.5 to 0.15 to 0.45%, and hence our methodology incorporated a procedure in order to study the effect of the bile salt concentration at 3 different doses, that is at 0.15%, 0.3% and 0.45%. As we increase the bile salt concentration and as time progresses, you see decline in the cell viability. Furthermore, it is also important to determine the effect of pepsin and trypsin on the viability of a strain. We all know pepsin and trypsin are the digestive enzymes which aids in the digestion of the protein, and the pepsin is present in the stomach compartment and trypsin is present in the small intestine. So in this experimental procedure, we have studied how the pepsin at varying concentration as well as trypsin at varying concentration will have an effect on cell viability. And this figure will show you as you increase the pepsin and or trypsin concentration from 0 to 10 grams per liter, there is a severe decline in the cell viability. All of these procedures are concentrated on evaluating the viability of a strain as it's being exposed to different compartments of human GIT. We started from mouth compartment, wherein we studied how salivary alpha amylase will have an effect on cell viability. Then we moved down to stomach compartment and we studied how the effect of pH will have an effect on the viability, as well as the effect of pepsin affects the cell viability. Moving to the small intestine, we studied how the ox-gal, that is the bile salts, affects the viability of a probiotic microorganisms, as well as how trypsin present in the small intestine will also bring down or will have an effect on cell viability. So having known how the viability of a probiotic microorganisms will vary with respect to different compartments, we can determine what is the viability of a probiotic strain that reaches the colon or a large intestine. Therefore, it helps us to determine what should be the initial dose of the probiotic microorganisms that is required in order to achieve 10 power 12 CFU per ml of the viable cells reaching the colon. Moving forward, in order to determine the other characteristics method, now let us discuss the organic acid production profile. What are organic acids? These are the acids which are produced by the lactic acid bacteria or a probiotic microorganisms as their metabolic end product. The examples of the organic acids produced by probiotic microorganisms include short-chain fatty acids. As the name says, these are the fatty acids of short chain wherein the chain length varies between C3 to C6. And the examples include acetate, butyrate, propanoate, and lactate. It is very much important for a researcher or an industry which is working on probiotic microorganisms to determine what are their metabolic end products, specifically short chain fatty acids, which are the organic acids, the metabolic end products of a probiotic microorganisms, and its concentration levels in a supernatant. Ideally, we use high-performance liquid chromatographic procedure for quantifying short-chain fatty acids, which vary in chain length between C3 to C6 carbon atoms. And the examples include lactate, butyrate, propanoate, and acetate. So as you see in our case, for the strain which we were using as a probiotic strain, was able to produce more amount of lactic acid in comparison to other acids such as butyric acid, propanoic acid, as well as acetic acid. Higher the amount of short-chain fatty acids being produced by the probiotic bacteria, the better health benefits it offers. The next characterization technique is the antibiogram. This is a laboratory test which describes the antibiotic susceptibility of a probiotic strain. Why this test is necessary is because it helps us to evaluate the safety of the probiotic strain. Some probiotic strains may carry antibiotic resistance gene, and this antibiotic resistance gene may be naturally present in the probiotic microorganisms, or it could be acquired from the other bacteria. Hence, it is very much in need to know if they are safe and they do not transfer these resistance gene to pathogens. The second point why it is important to do antibiogram is to determine its effectiveness during co-administration. In case if we give probiotics plus antibiotics together, which is quite common in a clinical setting, the probiotic should be resistant enough to survive if needed, or it should be susceptible enough to be eliminated if required. And in case if we are approaching for a regulatory compliance, one such example could be EFSA. So EFSA we have discussed while we had a discussion about functional foods and nutraceuticals introductory class, right? EFSA stands for European Food Safety and Standards Authority, as well as other regulatory bodies require antibiogram profile of a particular probiotic strain in order to get a market approval. Hence, antibiogram is also a type of characterization procedure which helps us to determine the susceptibility of your strain towards the clinically relevant antibiotics. This slide shows the summary of the susceptibility of the bacterial strain which we used in our lab. So as you can see, the organism was found to be sensitive to most of the clinically relevant antibiotics including vancomycin, tetracycline, ampicillin, chloramphenicol, and rifamycin and few others, and resistant to few such as polymyxin B, erythromycin, and few others. So therefore, this antibiogram, the laboratory procedure, can be viewed as a crucial tool in clinical microbiology in order to determine susceptibility or resistance of a strain towards a clinically relevant antibiotics. The next characterization method is the antimicrobial activity. What is antimicrobial activity? So as I said, organic acid profiling as a method of characterization, right? Because your probiotic strain is capable of producing short chain fatty acids such as lactic, butyrate, propanoic, as well as acetate. So these short chain fatty acids comes with varied health effects. One such health effect could be possessing antimicrobial activity towards the pathogens which are present in your gut, ecology or the gut environment. Therefore, antimicrobial activity is also a crucial laboratory technique which helps us to determine whether the supernatant or the postbiotic compounds produced by your probiotic strain will offer resistance against the pathogenic strains which are present in the gut ecology. So in this procedure, what we have done is we have done the antimicrobial activity of our strain against the pathogenic microorganisms such as Listeria monocytogenes, Staph aureus, Staphylococcus aureus, Bacillus cereus, and E. coli via plate method, and we were able to observe zone of inhibition in case if the organism was capable of producing the postbiotic compounds which offer resistance against these pathogenic strains. This slide shows the antimicrobial effect of the supernatant, which consists of the postbiotic compounds produced by a particular probiotic strain against the pathogen, and the pathogen used in the study is Escherichia coli. As you can see, in comparison to the control well, which comprises of just sterile phosphate buffer saline, and the crude extract showed a significant antimicrobial activity. This is one such the potent property which can be exploited in order to achieve a particular target health effect. Now let us assume you have you have been successful enough in isolating a probiotic microorganisms from a given conventional or a non-conventional source, and you have done a normal preliminary characterization procedure using gram staining as well as catalase test, and then adopted genome identification procedure using 16S RNA amplification process, and you know your culture belongs to a lactic acid group of bacteria. Post this, you have adopted a characterization procedure in order to characterize the probiotic microorganism trait, and you have come up with this organism in a powder form or a pill form or in a tablet form, or you can also come up with a fermented food by using this fermenting microorganism. Now, let us see what could be the potential health benefits that your microorganisms can be exploited to offer. The first health benefits of a general probiotic microorganisms can offer is the balance out good and bad bacteria in the gut, thereby help in maintaining the proper ecology, meaning it offers digestive health benefits. In addition to this, there are also few strains of lactic acid bacteria like we have discussed while discussing the history of a probiotic microorganisms such as Lactobacillus acidophilus. They specifically thrive on the milk sugar, which is the milk sugar. We know milk sugar is lactose, right? So thereby using the strains which have ability to ferment milk sugar, we can produce fermented dairy products using lactose degrading or lactose fermenting probiotic bacteria, which can be used as a solution for lactose intolerant people. The third health benefit could be they also regulate immune system. They strengthen the immune response by modulating inflammation, and they help prevent infection. The few probiotic strains are known to have better anti-inflammatory activity. They also influence mental health to gut-brain axis through the metabolites such as short chain fatty acids, neurotransmitters. Few probiotics are capable of producing neurotransmitters like GABA. They also modulate the neurotransmitter production like serotonin and dopamine, which helps in proper mental well-being, thereby reducing anxiety and depression. Few probiotic strains will also offer better digestive health benefits towards diarrhea in order to prevent and reduce the duration of infectious diarrhea. Few probiotic strains will also offer digestive health benefits. They help to maintain healthy vaginal microbiome, especially the genera which belongs to lactobacillus. They also prevent infections like bacterial vaginosis and prevent yeast infections. Few probiotic microorganisms have ability to modulate allergies and reduce eczema allergic conditions, as well as they are reported to reduce the food allergy conditions. These are the few health benefits that the probiotic microorganisms can offer in addition to offering better digestive health benefits. This slide shows the specific probiotics which can be exploited in order to achieve a targeted health effects. One such example could be Bifidobacterium genera. They help to prevent obesity as they are reported to influence metabolism and fat storage. The exact mechanism by which these probiotic microorganisms will offer specific health benefits we would be discussing in the coming lecture videos. The next example could be the Lactobacillus genera. They are reported to improve oral health by reducing harmful bacteria and promoting beneficial ones in the oral cavity. We have Escherichia coli. Few strains of E. coli microorganisms are available as a probiotic ones in the market, and they are reported to be safe strains, and they are used to treat osteoporosis by modulating gut calcium absorption and metabolism. We also have Bacillus LBP32, which is known to have antioxidant potential as they show ability to reduce oxidative stress via antioxidant mechanism. We have Lactobacillus acidophilus, which is reported to support skin regeneration as well as modulate local and systemic immune responses. We have Streptococcus thermophilus, which improves the nutritional status and is widely used in the preparation of the fermented foods as it aids in digestion as well as vitamin production. We have yeast strains such as Saccharomyces cerevisiae or Saccharomyces boulardii, which is reported to offer immune enhancement property. We have Lactobacillus rhamnosus, which improves digestion. So these are the few examples of the probiotic strains which are commercially available, which can be used to achieve targeted health benefits. This slide shows how we classify probiotic microorganisms which are available in the market. The probiotics are classified as monoprobiotics, polyprobiotics, and compound probiotics. What are monoprobiotics? These are the probiotics which comprises of just one species and one strain. Example could be having the probiotic capsule or a tablet or a powder which comprises of just one species and one strain, such as Lactobacillus rhamnosus. GG. Lactobacillus is the genus name. Rhamnosus is a species name. The strain name is GG. The monoprobiotics are the probiotics wherein you have the probiotics which comprises of just one species and one strain. Example could be Lactobacillus rhamnosus GG. The second category is polyprobiotics wherein the probiotic tablet, capsule or a powder, it is made up of one species but different strain. Example could be having Lactobacillus rhamnosus GG as the first probiotic microorganism. The second could be Lactobacillus rhamnosus. Instead of GG, it could be some other rhamnosus strain. So this is the example for polyprobiotic microorganisms. The third probiotic classification is compound probiotics, wherein you have more than one probiotic species and genera. Example could be having Lactobacillus rhamnosus GG along with yeast such as Saccharomyces boulardii. In addition, you can also have other bacterial genera like Bacillus subtilis or Escherichia coli. So these could be an example for compound probiotics. The probiotics are classified as monoprobiotics, which are made up of one species, one strain. Polyprobiotics, which is made up of one species but different strains. Compound probiotics, which is made up of different bacterial genera. So this is the probiotic tree, which comprises of different probiotic genera, the bacterial genera most importantly. However, we have the yeast genera such as Saccharomyces species. Other than this, everything else is a bacterial genera. The ones which are widely used is Lactobacillus species, Lactococcus species, Enterococcus species, Pediococcus, Streptococcus, Vicella species, Bifidobacteria species, Escherichia species, Bacillus species, Leuconostoc species, and yeast such as Saccharomyces species. These are the few examples of the probiotic strains which can be exploited for their specific health benefits. In previous slide, we saw how probiotics can be classified based on their composition, right? Polyprobiotics, monoprobiotics, and compound probiotics. Depending on the type of the bacterial genera, species, and the strain present, we can classify probiotics as monoprobiotics, polyprobiotics, and compound probiotics. In addition to that type of classification, probiotics can also be classified based on the genus level. What is the genus level classification? the classification which is made to belonging to a probiotic microorganisms to different genera. The genera which are most commonly being used include lactobacillus and bifidobacteria. So this classification is focused just on genera. The genera which belongs to lactobacillus, the genera which belongs to bifidobacteria, the genera which could belong to streptococcus, the genera which belongs to enterococcus. This is the genus level classification of probiotic microorganisms. We can also have species level classification. Within each genus, there could be different species. One such example could be Lactobacillus acidophilus, Lactobacillus rhamnosus, right? So, or Lactobacillus casei. So, this classification is based on different species. Under this, we have under Lactobacillus genus, we might have various species like Lactobacillus acidophilus, Lactobacillus casei, Lactobacillus rhamnosus, Lactobacillus plantarum. Similarly, even for Bifidobacterium genus or Enterococcus genus, we might have different species. This classification is based on genus. The next classification can also be done on strain. The most specific classification is at the strain level, not at the genus level, not at the species level, but it is at the strain level. Why it is at the strain level? Because the health benefits that the particular probiotic microorganisms will offer is strain dictated. That is why the most important or the crucial classification is based on strain level classification. The probiotic strains are identified by a specific combination of the letters or the numbers. One such examples could be Lactobacillus acidophilus. Lactobacillus acidophilus. Lactobacillus is a genus name. Acidophilus is a species name. Now, depending on the isolation source, you can name your probiotic microorganisms, such as Lactobacillus acidophilus La5, acidophilus 5. So meaning it could be a fifth culture isolated from a particular lab. Or you can give La10 depending on the type or the number of the cultures you have been dealing with. The next classification is based on functional classification. Here the classification is based on their intended function. Examples could be the lactobacillus, which offers immune support. Examples could be Lactobacillus acidophilus or Bifidobacterium genera, or the lactobacillus or the lactic acid bacteria, which offers neuroprotective benefits. So, the classification is based on functional attributes rather than general or species or the strain classification. Next classification is based on host specificity. The probiotics can further be classified based on the preferred host. So what do I mean preferred host? Meaning some strains are commonly associated with humans while others are used in animal probiotics. So this classification is based on human probiotics, meaning the probiotics which are used for human application, animal probiotics which are used for the application of animals other than humans. It can also be aquatic probiotics which can be used for aquatic applications. The next classification is based on mode of action. How do they act? So this classification is based on the microorganisms or the probiotic microorganisms for their ability to produce antimicrobial compounds, or for their ability to produce higher level of short chain fatty acids, or for their ability to produce antibacterial peptides. So this classification is based on their mode of action. The next classification is based on viability and formulation. On viability, we can classify probiotics as viable probiotics and non-viable probiotics. Viable probiotics are the organisms which are viable or live in nature, which when ingested, it offers health benefits. Non-viable probiotics, they slightly deviate from the FAO/WHO given definition of probiotics because they are non-viable in nature, and non-viable probiotics are known as the non-viable probiotics are known as paraprobiotics are known as paraprobiotics, which are not live in nature. However, when ingested, they might offer some or specific health benefits. The last classification is based on formulation. Formulation meaning the final end product type, which could be in the form of powder, it could be in the form of capsule, tablet, or in the form of functional food. So this is how we classify probiotics based on genus, species, strain, mode of action, viability, formulation, and few other type based on which we can classify probiotic microorganisms. Thank you. See you all in the next lecture video.